Good morning and good afternoon, and thank you for joining us for today's webinar. My name is Maria Hubert, and I'm representing Bristol Myers Squibb, and today I'm joined with Stan Kwok from AstraZeneca, who is co-leading Subvisible Particle Working Group under IQ Consortium, and we will be helping with the webinar. Today's presenter is Dr. Lindsay Crawford from Pfizer, and she obtained her PhD from Cornell University in chemical engineering with a research focused on targeted drug delivery of nanoparticle therapeutics. She then went on to do the postdoc at Washington University in St. Louis, where she focused on hydrogel scaffolds for delivery of regenerative medicines. From there, in 2016, she joined Pfizer, where she is leading particle group within the drug product design and development group in biopharmaceutics, pharmaceutical sciences. She is also people and project lead. Lindsay, we were very happy Lindsay could join a Subvisible Particle Working Group in 2020, but she already contributed to our work prior to 2020 when she helped us with collecting data, which you will see later today in the webinar. So thank you, Lindsay, for presenting today, and good luck.
Yes, thank you so much, Mario, and thank you, Stan. And with that, I think we can start with acknowledging the vast amount of scientists who have really contributed towards this work that we're going to present here today. And I especially will thank Mario and Stan for helping as well with the design of the slides and putting all the data together for this presentation so that we could share this exciting work with you all, as well as some of the learnings that we've taken away from this large study. Great. So today I'm just going to briefly show the background of where we fall into the IQ Consortium, where this particular piece of work kind of falls in. And then we'll get into the meat of the presentation with the introduction, our objectives, looking over the different techniques that we used in this study, and then getting into the results and then the conclusions we were able to take away and where our future work is leading us. Okay, so the Subvisible Working Group falls within the Biological Leadership Group. I won't read all the details here, but can certainly go back and read the details of what the Biological Leadership Group mission is. Next slide. And then these are the working groups that fall under the Biological Leadership Group. And then there you'll see the subvisible particles, which is where our Subvisible Working Group comes. So again, the bulk of our work that I'm going to discuss here really is derived from the past few decades where protein therapeutics as a whole have really continued to evolve. With this evolution, there's been a greater understanding in various analytical tools and characteristics that should be monitored to assess the product quality of these biotherapeutic products. Given the complex structure of many biotherapeutics and the very different mechanisms of aggregation that we might see from other traditional therapeutics, particle generation has been deemed an important characteristic to be studied. Subvisible particle characterization has been the subject of numerous studies. You can see a few examples here throughout the last few decades. And there has really been a significant amount of development in the characterization techniques and the understanding among the industry on how we can use these techniques and what those results mean to us for our products. The subvisible techniques, however, are limited down to a particle size of 1 micron. And in recent years, there has been regulatory interest in characterizing submicron particles, which we classify as particles less than 1 micron. Next. Unlike the subvisible particle characterization techniques, there is less known about the techniques for characterizing submicron particles for biotherapeutics. The technologies that we do have available to us are derived from other fields, but there's been relatively limited studies, especially in comparison to the subvisible techniques, on how we can develop and use these for biopharmaceutical applications. In addition to the lack of the understanding in these characterization technologies, there's also a lack of relevant data to understand what we may expect for submicron particle levels in both clinical and commercial products. Therefore, the Subvisible Particle Working Group saw an opportunity to collect this information and put into context the level of the submicron particles that we could expect across products from various companies. Additionally, this study can help us gain an understanding of the robustness of two of the more common methods that we've seen used throughout the industry for submicron particle characterization. And these two are nanoparticle tracking analysis, or also referred to as NTA, and resonance mass measurement, or RMM. In order to accomplish these rather large goals, we wanted to obtain both information on the levels of submicron particles in clinical and commercial products, as well as examine the robustness of the current submicron particle methods that we had mentioned previously. And to do this, it was decided that we could look at a particle size range of 0.1 to 2 microns and look at both late-stage clinical and marketed drug products. Each sample would be measured in triplicate, and ideally we would have multiple lots. This would allow us to assess things like robustness and accuracy of these methods, as well as capturing some sample metadata, such as maybe the sample type, whether it was maybe a protein, maybe a specific mAb or a fusion protein, the different package that it might have been in, a vial, maybe a syringe, as well as a variation in protein concentrations that we might see across products. And it also, this also allows us to look at the instrument performance across all these different variables. We also strived to harmonize our methods for data collection so that we could really assess the robustness from lab to lab, as well as just of the instrument themselves. And to further determine method robustness, we also looked at different size standards that we could use, as well as protein standards, to account for just variability in maybe the instruments or the lab-to-lab variability as opposed to product variability. Lastly, as part of this study, we also worked to identify and implement an anonymous data storage solution that then enabled various companies to provide products and data for a very comprehensive study in a double-blinded manner. The first technology that I mentioned that we were looking into was nanoparticle tracking analysis, or NTA. Nanoparticle tracking works based off the principle of Brownian motion, whereby a laser scatters light of the particles and the sample in the sample, and the particles are then essentially picked up by a microscope, and the movement of those particles is tracked over time by that microscope. The tracked movement is then correlated to size based on Brownian motion principles. NTA is capable of measuring particles in a size range of 10 nanometers to 2 microns, and concentration range of 10 to the 6 particles per milliliter to 10 to the 9 particles per milliliter. NTA is mainly used for nanoparticle characterization needs when the nanoparticles are known to be present in the sample, such as various nanoparticle therapies, or maybe for other types of nanoparticle applications that aren't in the pharmaceutical industry. However, there's some challenges that can arise if the sample does not scatter enough light to be detected, and that's just one challenge that you can face when you're looking at biotherapeutic products. Next. The second technique that I mentioned that we looked into was resonance mass measurement. And this is a technique where the sample is applied in a microfluidic chamber, and there's two different size chambers that can be used with this, a micro and a nano sensor. Within this chamber, there is a resonating cantilever, and as a particle passes the cantilever, it disrupts the frequency of that resonance. This disruption in the frequency is accounted for, and that is correlated to a particle size. The concentration range is similar to NTA for what what can be measured on RMM, in that it's 10 to the 5 particles per milliliter to 10 to the 9 particles per milliliter. And again, I mentioned that there's 2 different sensors that can be used, and they carry different size ranges. I will note here, as it does become a little bit important later, is that the volume that's measured for this technique is very small, as well as the volume measured for NTA. One more thing. One other additional feature of resonance mass measurement is that it does have the ability to distinguish between buoyancy of particles. So it can distinguish between a positively buoyant or a negatively buoyant particle. And this can be a very unique and interesting feature of this technology. As you imagine, if we had a container closure that was like a pre-filled syringe where we would expect maybe more silicone oil particles, you might be able to look at those different populations compared to, say, protein particles that would be negatively buoyant. Next slide. So now I'll start to talk about the different results that we've seen. And I want to start by the focus on the standards that we used. So first standard was a size standard, and this was a polystyrene bead standard that was used. And we used both NTA and RMM for this. On the left-hand side, we have the NTA graph, and on the right-hand side, we have the RMM graph. I want to point out that on the y-axis for NTA graph is in nanometers, where the y-axis for RMM is in micrometers. So there was 2 different size standards that were used for these technologies based on the size range that they could accommodate. The concentration, or sorry, using the NTA technology, we can see the mean particle size of the standards was within 10% of that 200 nanometer for labs, and the labs are down on the y-axis, for labs A, C, G, H, L, and M. But they were outside the range for labs F and K. The difference in the results suggests that there is an overestimation in particle size during measurement or changes in the particle standard. Since it was moderate and only seen in 2 of the 8 labs, it's not considered to impact further interpretation of the data. When we look at the RMM technology, the mean size of the particle standard is within the 10% of the 1 micron standard for all the labs. So we looked at the size accuracy with the polystyrene standards, but to really assess count reproducibility, we wanted to look at a protein control standard. And this is due to the fact that what we see for protein controls and what we see for protein therapeutics can be quite different than a round polystyrene bead, as they have different optical, physical, and morphological properties than protein particles. So similar to what we saw with the size standards, we have different labs that assessed the samples, and this was a stressed BSA sample, and it was the same stress material that was provided to all the different labs. On the y-axis here for these graphs, we actually have particles per milliliter. And for RMM, this is specifically at 0.4 micron or greater. From the NTA data, what we saw was that the average RSD between all labs was 36%, and for RMM, it was 20%. This is the average between different labs with similar instruments and the same samples. The values are comparable to other subvisible particle techniques when they were also compared to multiple labs. What's worth discussion here is that the difference in particle concentration between the 2 common submicron technologies spans almost 2 orders of magnitude. This is partly explained by the size ranges which these technologies operate in. NTA is shown here for particles greater than 0.05 microns, where RMM is shown for particles greater than 0.4 microns. And we know that at lower particle sizes, you tend to have higher concentrations. However, what isn't shown here is that when the NTA data was adjusted to particles greater than 0.4 microns, there was still in 1 order of magnitude higher with RMM. And this just suggests to us that the technologies can't be considered orthogonal techniques, but rather they perhaps complement each other as they operate under different principles and they're leading to different concentrations. So when we start to look at the different products that we considered in this study, we wanted to point out here that we had 52 unique products and 62 total samples. There was different lots of some of the products. And these products spanned in the variability of what the container closure. So it was either a syringe, a vial that was a liquid formulation, a vial that was a lyophilized formulation, or an other type of container. We looked at different protein concentrations, anything from 5 milligrams per milliliter all the way up to 100 milligrams per milliliter, with the majority of these samples being located between 5 and 50 milligrams per milliliter. And greater than 70% of these samples were tested by NTA and RMM. This allowed us to collect a significant amount of particle data and size data from NTA and RMM and capture it in the database. The focus of this presentation, however, is on total particle counts in the clinical and commercial products. So using this large data set to compare the NTA and RMM, we can actually take away a few conclusions. The graph on the bottom left shows a correlation between particle counts with RMM and particle counts with NTA. As first noticed with the protein standards, the NTA counts are 2 to 3 orders of magnitude higher than RMM for particle concentration. When we compare the counts between samples with both techniques, there is a slight correlation, which was a Pearson correlation coefficient of 0.39. However, it is weak. The weak positive correlation suggests that the techniques can be somewhat comparable for determining changes between samples, but the order of magnitude difference suggests they cannot be compared for directly for particle concentration. So we can imagine that these technologies could be used if we had a control sample to compare against for various, scenarios. However, we can't compare them directly by saying the RMM particle counts is comparable to the NTA particle counts. Another difference between these two techniques, as noted here, is the mean particle size that was detected. RMM detected over double the size of NTA for the mean particle size. This is most likely due to the optimal size range with which the two technologies can operate in, with NTA being able to detect a lower particle size and RMM being able to detect a little bit bigger particle size. Again, this points to the technologies that they're not orthogonal, but they're rather complementary to each other. So similar to what was done with the different standards, the polystyrene beads and the stressed BSA protein particle standards, the robustness of NTA and RMM was also analyzed for the various different products that were used in this study. Each of the techniques was run in triplicate again for various samples that were of varying protein concentrations from 5 to 100 mg per ml. And these products were either in syringe, a vial, or another container. And then they were also further divided between either liquid or lyo products. As we can see in the graphs here, and again, NTA on the left, RMM on the right, the percent RSD for both NTA and RMM was much higher compared to what we saw for the protein standards or what we saw with the size standards as well. And this is also much higher when we compare it to existing knowledge of subvisible particle techniques like Hayek and MFI that are very well established for biotherapeutic products. The average RSD for NTA across the samples was 51%, and for RMM it was 73%. But the ranges for these was 1 to 120% for NTA and 2 to 200% for RMM. There was really no correlation either with the particle concentration and the RSD, as regardless of what concentrations we were seeing, there was still that large variability. So to further look at the robustness with the clinical and commercial products, It has been suggested in literature that poor repeatability of these techniques can partly be due to the small sample size and then sample volume that is actually measured with these techniques, which can then lead to poor sampling efficiency. The sample measurement volume being low means that there is actually significant extrapolation factor applied to determine actual product concentration. And this extrapolation factor can then lead to poor repeatability and precision. This notion combined with the results of this study suggests to us that repeatability is likely product dependent, and it may really depend on factors such as the actual particle concentration in the sample, the matrix of the sample, among other potential factors as well. These results suggest an inherent limitation of submicron particle counting techniques when it comes to comparing between products. One of the other major aims of this study, aside from determining the robustness of various submicron technologies, was to evaluate the range of submicron particles that we could expect in various late-stage clinical and commercial products. This looked across syringes, vials, liquid and lyophilized products again, and at those various concentrations. The data from NTA shows the results spanning 4 orders of magnitude, and for RMM, we're spanning 3 orders of magnitude for the various different products. There's really no clear visible trend between the submicron particle size and either the container type, the concentration, or if the product was a liquid or lyophilized products. Some products had higher values of NTA measurement than the instrument could actually detect, and they had to be diluted prior to analysis, which applied another multiplication factor for those samples as well. So a major takeaway from this particular analysis in this slide is that the values of submicron particles are going to be quite variable for even well-developed products. Next. So to further evaluate any correlation between the various metadata that was collected in this study and these submicron particle values using these 2 technologies, statistical analysis was also done. The results did show no statistical significance of the factors study. There might have been weak correlations here and there, but there was really no clear trends or significance that could be pulled out of this data. Given the unique feature of RMM is distinguishing between positively buoyant and negatively buoyant particles, it was also interesting to see that there's no differences in positively buoyant particles between syringe and vial, where we would expect more silicone oil particles in a syringe compared to a vial. This could also suggest that silicone oil particles might be just more prevalent in particle sizes greater than 1 micron, or it could be a factor of the technology, but most likely due to the fact that they tend to be more subvisible particle sizes. So now if we go through some of the conclusions from this large amount of data that we kind of quickly went through, we did analyze 52 unique clinical and commercial protein therapeutics that looked at 62 dosage forms for the submicron particles by NTA and RMM. The particle concentration in therapeutic products, we do have a range from 10 to the 7 to 10 to the 11 particles per milliliter for NTA and 10 to the 4 to 10 to the 7 for RMM. Again, there was really no significant differences in the submicron particle concentrations as a function of any of the various different metadata that we collected, whether it be sample type, package type, or the concentration. The levels should really only be compared with a given technique and cannot be used to predict levels by other counting techniques. And again, the levels can really be compared between a control and a maybe stress condition or a control and another type of condition. However, we cannot compare between products. The results obtained by both NTA and RMM had high variability compared to our well-established subvisible techniques that we're used to using, such as light obscuration, or we refer to as HIAC often, and flow imaging, which can also be called MFI. The NTA and RMM characterization techniques may provide relevant submicron data during drug product development, but they are not appropriate for quality control related testing. So all the results and details of this work were published in an article in January of 2020, and the reference can be found here. So I welcome anybody who has further questions or wants to see, even more details of the data to go and look this publication up. So the conclusions from this submicron particle data has led our working group to pursue questions of the changes that we might see in subvisible particles depending on the similar factors considered and presented in this study. So that is the focus of some of our ongoing work. We're aiming to compare the different subvisible techniques in a similar manner to the submicron work presented here, and looking at some other relevant factors for submicron particles compared to for subvisible particles, as well as another focus group looking at silicon oil specifically. Next slide. With that, we'd like to acknowledge the IQ for providing the support for this work, as well as allowing us to give this webinar for everyone today to really send the message of the work that we did and the learnings that we had from it. Next slide. And with that, myself, Mario, and Stan would be happy to take any questions that anyone might have.
Thank you, Lindsay, for the wonderful presentation, overviewing the work, collaborative work we did between the various IQ members to put this paper together. And again, we do have a few questions from the audience. And again, if anybody have anything else to ask, please feel free to roll them in as well. And I want to mention that again, the slides and the recording of this video will be available later on for everybody to share. Okay. Now, with that in mind, I will start going over some of the lists and then some of the questions that people have asked, and then we'll talk about it in our panelists. A lot of these questions are related to the techniques, so which is great. We have a lot of technical interest in some of these technology, but we also welcome questions on kind of strategies and thinking as well. So the first questions that is asked is, how are these two techniques selected as the techniques to study? Why were NTA and RMM used?
Yeah, I can start with the answer of that question, and if you have anything else, Stan or Mario, to add to it. But I think these were the two most common techniques, especially at the time, that were being applied to biotherapeutic products for submicron particles. And so I think due to the I want to say popularity of these two submicron techniques between labs and into the industry, I think that's probably the rationale for choosing them at that time.
We have to agree, when we started this work, we polled all the team members contributing from the various contributing members in pharmaceutical as well as biotech companies. These were the most prevalent techniques in the analysis of submicron particles in their labs at that time. As well, both of these techniques can study particles in the submicron size range, providing both quantitative counts as well as size information. There are, of course, other technologies and other methods that can kind of give you characterization of your drug substance or your drug product, but we were focusing on size and counts in qualitative manner. Again, both of these mechanisms measure submicron particles, but also an orthogonal mechanism. The technology is different. So we're able to kind of get the same product attributes, but using two different methods, so we can also compare the technology as well. So the next question, which is related, but not exactly the same. The question is the nanoparticle tracking analysis technology the same as DLS technology? And then would the synthesizer also be useful in the size range?
Yeah, so nanoparticle tracking is slightly different than the DLS. While they do both scatter light, DLS doesn't typically capture concentration data, so it doesn't actually count the number of particles within the sample. It more gives a size distribution and isn't able to really provide the actual concentration of particles within that size range. So they do differ in that a little bit, and that's very high level of their differences. But you can certainly get a size distribution and, look at a polydispersity when you look at DLS, but you wouldn't get the concentration information.
Lindsay, this is Mario, if I can add just one sentence. Basic difference is they are both based on Brownian motion of particles, but NTA is measuring single particle at the time, while the DLS is measuring bulk or the cloud of particles at the time.
Yeah, so we have a couple more questions that's for the physics audience in the background as well. So the question they have is, why is it called positive and negative buoyancy in RMM? So, and I think this is a question that Lindsay have alluded to during the presentation itself, and I will gladly take this question. So when you're doing product development, when you're moving from, for example, vial to pre-filled syringes or cartridges, often you encounter devices which have higher silicone oil content. The silicone oil droplets in the cantilever chamber of the RMM is generally have density that's less than the buffer. So what this will do is it will produce a positive peak in the frequency of the traces and reduces the overall masses of the sensor. But behind the physics of this is that essentially when you do RMM analysis of your product, you can detect things that are potentially different from your proteinaceous particles. So the silicone oil will produce positive peaks and your proteinaceous will have negative peaks. So this gives you an additional level of information that you can see when you're doing your characterization. And I think we'll wrap up some of these techniques and methods in the next questions also. Okay. The next question I have is, what kind of size is calculated in each of the methods? Is it hydrodynamic radius for NTA and geometric size for RMM? Mario and Lindsay, do you want to take that one, or do you want me to say something about Stokes radius and calculations?
You want to go ahead?
Yeah, sure. So again, we have a lot of physics on the background. Okay, so when the NTA, again, it does use Brownian motion and the kind of diffusion coefficient to calculate your size. And it would use the model of a hard sphere that diffuse the same way as a solid. So again, essentially the NTA does model through what I call a hard sphere hydrated radius and provide hydrodynamic radius. And then if RMM is kind of a buoyancy equation, so again, it is more geometric size. So there may be slight differences, but sometimes they're used synonymously for our modeling. Okay. I think I may skip over some of these other questions about what I call most of these techniques in general. In the appendix of the publication, there is a review of the various subvisible and submicron particle size characterization methods that's spanning from these nanometer range to microns range or higher submicron range. And the audience can look into this regarding what's the best technique and the methods for you to characterize the range of your interest. And there's also an appendix for some of the best used practices. For example, the replicates you should take, the amount of dilution you should use for these methods. Okay. All right, this is a good question. This is more QC related. In the current industry, is there any submicron practical methods could be used for QC release testing for nanoparticles in the 50 to 150 nanometer range?
So I'm interpreting that as, oh, go ahead, Mario.
Yeah, Lindsay, you can then follow up. So yes, one of the goals of the work was to answer these specific questions. So we concluded these two particular techniques, whether it's NTA or RMM, are not robust enough to be used in the QC settings. But if the person is asking if there are any other techniques which could be used in the nano range, obviously DLS technique might be robust for that purpose, but we focus on techniques which are particle counting technique. So DLS technique, as it typically used, is not particle counting technique, it's particle sizing techniques. So in particle counting techniques, we are not aware as this working group of any technique in submicron range for particle counting purpose that would be suitable for QC environment. Go ahead, Lindsay.
Yeah, I was just going to say, I think if the scope of the question is therapies that are nanoparticles and you're trying to, you know, to look at technologies for that, then yeah, I agree, Mario. DLS is typically the technology. But again, it doesn't do concentration.
So the next question is a little bit more what called general and forward looking. So I think this will be kind of good for our audience to kind of chime in on this one as well. So the first one is, what was the impetus for FDA's suggestion to evaluate submicron particles? And is this expected to be something that we monitor in our drug products in the future once methods are more reproducible? So I first want to make the disclaimer that we are not FDA, so we would not what I can say on the behalf of the impetus, but some of it has come from our experience in drug development and the questions and engagement with regulatory agency. But maybe Mario and Lindsay can also share their thoughts as well.
Sure, Lindsay, go ahead.
Yeah, again, like Stan mentioned, we're not the FDA, so we can't speak to the clear rationale of why they may have had, you know, that question. If you do think about, you know, protein aggregation in general, you imagine that as proteins aggregate, they change in size, right? They range from a monomer to a dimer to a ligamer, and then, you know, they go to something that's nano-sized, to something that's subvisible, to eventually potentially something that's visible. So I think if you just think of that size spectrum, you can see why you may want to consider it. However, you know, looking to see if there's actually a correlation between the different sizes of particles and potential product impact, I think that's still, you know, not something that's a clear correlation.
And if I can add, so one of the reasons why we look at this submicron range and why people also looking at subvisible range is implication of potential relation between particle counts or particle concentration or potential immunogenic response. So that was one of the reason why the submicron particle concentration was a hot topic a few years back, when there was suggestions that these particles, like other particles, might be related to immunogenic response.
Yeah, so I want to chime in as well. So again, when we look at particle characterization and analytical methods, sometimes things are bucketed into, oh, this is a USP requirement, this is a critical product quality attribute like HMWs and aggregates. But holistically, the whole topic should be kind of gauged more. Again, how do my products degrade, and what are the degradation pathway that leads from formation of dimer, trimer, and higher order aggregates to the assembly of these submicron particles, to even subvisible particle to visible particles. In the sense that how we triage this differently is really kind of more patient-centric, understanding the risk to your product and what are the mitigation. If we can have early detection of your products can be susceptible to certain risks, this will really help the drug developer. And again, linking this to immunogenicity, the group will continue to explore in this realm as well, linking patient centricity to potential immunogenic events and how we can avoid them. And one way to moving forward, as we have alluded to, is like potentially look at silicone oil. The silicone oil being positively buoyant particles, how do we understand this, the characteristics? So there's a lot of work that's ongoing, and I'm very excited. But the idea is to create safe, good drug products, then sharing the knowledge between the different consortium to be collaborative. So go to a couple more questions. This one is a little bit more clinical in use in nature, and maybe we can also share this experience. Any assessment done on the impact of 0.22 inline filtration on the submicron particles?
That wasn't part of this study particularly. Yeah. I don't know if Stan or Mario, you have any experience with anything else in this space?
Yeah, maybe I can start. And yeah, so with the recent kind of revision of the USP chapter, the 787 chapter, there is some there are texts in there showing that you have to have some control over your product and your admixture as well, right? In addition to releasing to there. So there is description of using this filter. Obviously, the use of a 0.22 micron or 220 nanometer inline filter can reduce your submicron subvisible particle load. but it is up to the sponsor, I think, to really understand and characterize your products from end to end, from during manufacturing, your risk of substituted particle, to kind of your dilution of your potentially your admixture. So yes, using of a filter can reduce your particle load, but you have to understand how your particle can form when things are diluted. So it's kind of a more end-to-end kind of controlling and understanding of your particle risk.
So as Lindsay suggested, in this particular study or this particular work, we didn't look at the effect of filtration on population of particles.
Yeah. Okay. All right. So this is a good question. What are some of the other newer technologies being considered for future studies?
I think, you know, when you look at other technologies within the submicron space, there is the submicron capabilities in SBOS, the, you know, FlowCam Nano has come out now, and those are just a couple that I can think of in the submicron space that people are starting to use. I don't think we have any particular plans yet in the Subvisible working group to look at any additional submicron technologies, but those are certainly ones that I've seen come out, you know, in the field in recent years. Is SBOS, I think it's FX Nano, and then the FlowCam Nano.
Yeah.
So yes, go ahead, Sam.
Oh, sorry, Mark. So I also want to mention that the technology advancement is not only in what I call hardware and technology, but also in the software of how these particles are characterized as well. I want to add that there's also one of the technology that's very promising. Spherics have a total holographic characterization. They make these really cool holographic diagrams, show how your particles flow through microfluidic channels. But in addition to what I call hardware advances, as Lindsay have alluded to, kind of the nano flow cam, we use the kind of more oil immersion microscopy techniques. Companies are coming up with new technology and artificial intelligence to characterize these subvisible and submicron particles, how to turn difficult to analyze particle images, morphologies, things like effective circular diameter, as well as contrast, into what I call a subvisible particle profile, how to characterize all these. So it is an exciting field of evolution, and we're looking forward to kind of more holistic and advanced characterization of our particles and how to match it to product quality attributes. Mario, do you have something to add as well?
No, I think you covered it well. Thanks.
Yeah. So again, it's just very exciting. Again, the working group is working with these partners, trying to adapt these technologies and learning. And hopefully we want ideas to build a complete toolbox so you can understand your particle risk from the very, very small particles, from aggregates, all the way to visible particles. So again, these kinds of information are very useful for both what I call benchtop analysis to manufacturing as well. So again, linking to really like product quality attributes in QTPP for patient safety and development. Okay, so I have a couple more questions and regarding to the kind of the immunogenicity aspect. Does this range of particles contribute to the immunogenic response from one of the audience?
In my opinion, I think the answer is we don't know. I don't think there's really like been solid evidence on the impact to immunogenicity that the nanoparticle size or, you know, the submicron size particles can play.
Yeah,
So.
that's Lindsay. I think that's a very challenging question for us to link.
It
There are
is,
publications showing what I call correlation, or what I call a publication kind of bring this interest into place. But I think a lot of it is really depend on your understanding of your product and during your development, what do you see? So again, this is another area we want to explore as a group as well. What are the clinical outcomes that can potentially relate to, you know, particle events or particle susceptibility? Again, that's a very tough question, but again, the more we understand and the more we share and get input and buy-in from other companies and the experience, the more we can develop safer products for our patients. Mario, do you have anything else to add? I do want to say that thank you for the audience for some of the wonderful comments and the comments on Lindsay's presentation. Thank you so much. Again, the audience is really receptive. And again, thanks, Lindsay, for all the work that's going into it. Maybe we'll have time for 1 or 2 more questions, Mario, before the wrap up.
Sure.
Yeah. Okay. Do you think this is going back to something a little bit more technical, which is probably in our wheelhouse of what we can do? Does the variation in viscosity from sample to sample impact the measurements?
I know specifically with RMM, viscosity can impact the actual like measured volume. So I would imagine that that could play a potential role with that measured volume. And with NTA, I'm not as sure. I don't know if Stan or Mario, you have insight into
Yes,
viscosity and
so the
NTA.
viscosity impacts both of the measuring techniques, but viscosity is one of the parameters which we inputting prior to measurements. In NTA, the viscosity plays important role too, because the more viscous, the less Brownian motion,
Right.
or the slower Brownian motion. So yes, viscosity is important for both techniques, and therefore we had to follow the recommendation for the viscosity range provided by the instrument manufacturers.
Yeah, so this is a good question. So again, with the data that is shown and from these marketed and late phase product, you do see a wide plethora of what I call size range that are being used in kind of the late phase clinical and commercial product. So the question I ask is, what with the large variability observed in the results, is the regulatory agency satisfied with the technique? So again, I want to preface that we're not the regulatory agency, so we cannot speak on their behalf. But I think one of the power of this paper is that with the benchmarking, you understand it depends on the product, your presentation, your configuration. You can see different levels of submicron particles for the various products that have used and have been in the clinic have been in the clinic and been used. So this is kind of the first benchmark of what are some of the levels and ranges we can see and what are the particle types that we can see for this. So I think that's a value added for benchmarking paper like ours. Lindsay and Mario, if you have something else to chime in.
Nope, I think you covered it.
Okay. Yeah, and again, I think that this is kind of a first step of doing some of this work, and we're very glad for the engagement and the audience member to come in. If you have anything to want to participate or interest in the IQ work, but the idea is to start the conversation so people can continue to explore. There was one of the questions from audience about with the submicron particles characterization, right? Are we going to measure subnanoparticles, and what information can we get? And when new technology evolved to look at smaller and smaller particles, how are things going to evolve? So I'll let Mario and Lindsay answer this tougher question.
Yeah, I let the Lindsay to answer the tough question, but I was going to ask Maya Leah. Maya Leah, could you please bring on the screen the slides like the number 2, where we list the members of the working group so people can look at it one more time, and if they are interested in this topic, they can also follow up with their particular company representative.
Yeah, so if I'm going to reiterate the question to make sure that I understood it. So it was about how we can use these submicron technologies, and with new technologies coming out that maybe can look at even different size ranges, how we kind of apply those. Hopefully that's the question, because that's the one I'm going to answer. So I think if you just in the space that with the understanding we have now, I think what we can really look at is the changes that we might see during development and various different studies, whether it be maybe stability or some sort of study during development. I think you can look at the changes that you might see, and when you compare them to the control, and that's a kind of useful way to use these submicron technologies as they are now. And as they further develop, I think forums like this where we have the IQ Subvisible working group, these are the great places to really look at how they develop and how they can be applied industry-wide, right? So I think that would kind of be my answer to the reiterated question that I hopefully reiterated correctly.
Yeah, so Lindsay, I think for me, it's always good to know more about your product. The more you know your product, the more you can mitigate certain risks, and understanding these risks early kind of de-risk your whole, what I call, development process. Again, there was an audience mention about, the use of filter and clinical mixture. So knowing your products and having these tools to understand these different things is going to be really helpful for us to develop new drugs and mitigating new risk. And again, Mario brought up an excellent point. This is a work for many, many people who did a lot of hard work and also contribute data from their own commercial and late phase products. So these are very, very valuable data. And we want to thank all the members that have put into really hard work for this, past and present, and maybe future as well, to kind of help, scope it, what I call help sculpt the way that particles, subvisible and submicron, and even visible particle, how are they going to be regulated, characterized, and understood. Mario, do you have anything else to add? We're running close to the top of the hours.
Nothing to add. Just thank you for Alia for organizing the webinar. Thank you, Stan. Thank you, Lindsay, for presenting and helping with the webinar. And thank you all participants for active participation and dialing in.
Yes, thank you so much for the opportunity, and look forward to